For the nuclear DNA, there are 8 steps. So we're going to go through each of these individually. The first few will also be used for mitochondrial DNA testing. And it's these final 4 that are going to be different in just a little bit when we talk about the mitochondrial. Okay, so let's talk about nuclear DNA analysis first. So first is that we need to collect our DNA sample. So of course we can find it in body fluids. Everyone knows blood, saliva, semen. Those things contain our DNA. But DNA can also be found on any object that is touched, licked, or worn. So you can have clothing, anything that has sweat in it. You might see in movies where, I don't know if you guys saw the movie Red Sparrow. It was not very good. But they tried to frame someone. So she got a glass that he had drank out of, and they used that DNA to plant at a scene so it seemed like he was at that scene. Okay, so you can use anything that was touched or licked to get your DNA on it in order to be tested. Almost anything can be swabbed. So swabbing is the most common way to get that DNA from an object into the lab in order to be tested. So here you'll see when they do a DNA sample from your mouth, that is what our clients who are convicted of a felony offense will have to give a DNA sample. They will always do the mouth swab because it's the easiest, least intrusive way to get the sample. So if you have a piece of evidence at the scene, you can either put the entire object into evidence to be tested, or you can take that larger object and cut it so that way it's a smaller object to be tested. All DNA samples have to go through this chain of custody properly, right? It has to be documented. Every time they pick up a piece of DNA, they do a swab on an object, it has to have the case number, the date and time it was collected, the description of the item, and where the item was collected. That is going to be extremely important. It has to be collected properly. So you ask yourself, was the professional wearing latex gloves? Was the object, or where was the object collected? Where was it swabbed? Did the sample properly dry, and should 2 or more swabs have been used instead of 1? Some of these questions rest on the technician or whoever was at the scene collecting this evidence. So this is very subjective. So if someone collected a piece of evidence, like the gun, for instance, and they decided they were only going to swab the trigger, that might not be the place where a good DNA sample is from, right? You may have wanted to get the butt of the gun, swabbed and tested. I know in one of the other schools, I think it's at Louisville, they had a sweatshirt put into evidence, and they used 1 Q-tip for the entire sweatshirt. So think about, are they going to get a good DNA sample? How much of that sweatshirt was actually passed over with the swab? One thing might be if the hood of the sweatshirt or the lining where it would touch your body, that might have been a better place to get a DNA swab rather than a sweatshirt that might have a shirt underneath it and you're not getting that sweat, or you're not getting those skin cells onto that sweatshirt. So those are things you need to think about when you have a DNA sample. Where did it come from, and could it have been done better? All DNA samples have to be handled and stored properly at every single stage. Once it's collected, one very important thing is to have a serology test done as well. So if you don't know exactly what it is, it's extremely important to figure out, is it semen or is it saliva? That can change your theory of the case. It can change how you're handling the case. It can change a lot of things if, for instance, it was saliva rather than semen. So that's the difference between perhaps oral sex and a rape charge, right? The second thing they do after they get this DNA collected is they have to take the DNA out of the cells. So there is a 3-step process to this. This is extremely simplified. But first is the lysis. They break open the cell. The precipitation, they separate the DNA from the other cell components. And then the purification, they isolate this DNA. So depending on your sample and how much of that DNA you can take out of that, they're going to use different chemicals, detergents, heat in order to get that DNA out of that sample. Once you have the DNA, you have to determine its quantity. So quantification, right, is determining the concentration and the purity of your sample. The measurement that they use is picogram. The super interesting part about this is how small we're talking about DNA. So if you think of a microliter is 1/1,000,000th of a liter of soda. A picogram is a 1/1,000,000th part of a grain of sugar. So one human cell has about 6 picograms of DNA, whereas a fingerprint has about 100 picograms of DNA. Like, that is extremely, extremely small. Now we get back to this amplification, right? We get the PCR. We're making copies of this DNA segment using heat and cooling in order to make these copies. And this is tested at very specific locations. So they are trying to say, this piece of DNA strand, we're going to look at this specific area. Electrophoresis. So this is taking that DNA now and sorting it by color and by size. So if you see this picture, it might be hard to see, but these are mama chickens and baby chickens. This was the best illustration that I could find where everything is divided by color and by size. So once it's divided by color and size, it's loaded on the front end of one machine, and the negative electrodes will kind of pull it through to the positive side, and this is where we're going to get our DNA results. And then you end up with this mess. So after it goes through the machine, you're going to get this blob. This is what is then used by the lab technician to interpret the results. It is extremely important to get this in your cases if there is a DNA sample, because the interpretation can be very subjective. A DNA lab technician might find something that another expert might look at this and say, no, they interpreted this incorrectly. By getting the raw data, you have that opportunity to give it to another expert so they can review the work of the first technician. So once the results are actually filtered, you're going to get this graph. And this is just a little bit more complicated than the graph that we saw before. But it has the same loci, the same locations. The alleles are what we're trying to interpret. And the number on the top here is how many are found from each parent, right? The strength of the allele, meaning how high these are, just means how good was the sample. If you have a lot of a sample, you're going to see higher peaks. If you have maybe a smaller sample, the peaks might not be as high. So it just means how much of that sample was there.